Hi everyone, tonight we are going to talk about the organogenesis direct from watermelon cultivar explant. I am Carla and this group is conformed by
Jorge Reyes, Kelly Gómez,
Luis Quesada,
José Gabriel de León.
Now we're going to present you our topic. Watermelon citrullonatus is a first-timer bean annual crop with sweet-tasting fruit that is often consumed as a cool dessert. The species originate in tropical Africa. The fruit of watermelon varies in size, shape, green color and pattern, flesh and seed color, and maturity date. In light of both its importance and susceptibility to disease, the watermelon is an excellent source for improvement through genetic engineering. To begin, we need to use the proper explant type because it's an important factor in successful organ regeneration. Not all explants from the same plant or cotyledon possess an equal regeneration potential. In watermelon, the best explant source is a cotyledon of in vitro germinate seedlings. In this experiment, a student will be culturing explants from different regions of the watermelon cotyledon to identify the best explant for adventitious shoot regeneration. Now, my classmate Luis Quesada is going to talk about the first step.
Okay, I will talk about the first step, that is the preparation of the medium and the planting of the explants. This is a simple step because we need just an explant from the mother plant, and we will planting this explant to have a new plant. This is the initial part of this experiment. First, after 5 or 7 days in the germination medium, they have germinate embryos, and they have 2 or 5 centimeters, and the vitroplantel have their cotyledon are still together, which they are in better condition. The vitroplantels are removed from the culture basils and transferred to sterile petri dishes for the dissection that they need to convert one plant to 4 plants. Once they have been removed from the root system and a half of the hypocotyl, the vitroplantels can be more easily managed. The cotyledons are removed by swallow cut widthwise between cotyledon, 1-2 millimeters near the points of sticking with the hypocotyl. Once they are removed, the cotyledons should be cut as shown in the diagram. Depending on the experiment, cultivate cotyledons abaxial side 2 in petri dishes containing below 25 milliliters. Read the regeneration medium. Seal petri dishes with paraffin. Identify the transfer for them to the grow chamber.
Hello guys, how I said at the beginning, my name is José Gabriel, and now I'm going to talk about the second step of this group that we are preparing. This presentation is for you because we want you to do a good job in this experiment we are doing. After 3 weeks of induction in cultural medium, the explant of the 2 varieties regeneration shoots, the adventitious buds were subcultivated for elongation a measure of root. That is the step that is going after this step. So in culture medium, that is named as Murashine and Scud. Hormone without BAP. Once the seed germinate and expose the yolk, we proceed to remove the cotyledon leaf. What happens when we remove the cotyledon leaf? Or we to proceed to cut the apical bud with a length of 2 centimeters, which it transfers to MS medium supplement with 1 milligram of 6-bent somelopurine that is named in smaller BAP. So to induce shoot formation, we do that in that medium. This activity was performed in 4 weeks. On average, that's an average that this occur in 4 weeks. For that reason, that's the second step in which we want to induce the shoot formation. So, how I said that this activity will perform for 4 weeks on average. So, which it corresponds to the time when the plant have exhausted reserve culture medium. And in this second step, we also proceed to the multiplication of our plants, and in that case we get from one plant that we have at the beginning, we proceed to divide using a strict control of no contaminate the medium or the plant we are using, even the skill we have to cut and divide that. We have to remember that when we do something like that, guys, we have to change the medium, because in the first step, if you remember, we use a medium without seed, because we want that the seed have a vigor and induce on it shoot formation. That is what we have right now in this step, and for that reason we change the medium and using cytosina. The cytosina is the best hormone to induce a shoot formation. And now my partner Frank Ellis is going to talk about the root formation, induce the root formation. And now I'm going to give him the time. Thank you.
Good morning, good evening, for continuing with the presentation of the organogenesis of the reproduction of the watermelon. I'm going to mention the step number 3, that is the produce of the root of the watermelon. This step is promoted for the housing. This is the best piece hormone for produce the roots in the plant. This begin 3 or 4 weeks after of the multiplication of the the multiplication of the plant. In this time, the plant needs to be 2 or 3 centimeters long, and the sheaths are ready to expand it and remove to the new culture of the medium. The apex of outbreak, the first 2 leaves are the cotyledon, and this outbreak to transfer to the magenta bulbs that contain the essential nutrient that can promote the root. The plants remove the necrotic of the root, and so you can to push to the new medium. And in this time, this is clean with ethanol of the ethanol in the 50% and for 10 seconds, and submerging them in hypochlorite of sodium for 10 minutes, and this concentration helps to eliminate to delete the bad things of the root. And for this is used the steady water. Performing this step, rinse water which it was proposed by rooting treatment, and this produced was performing within in a laminar flow. This is the best. This is the most important in this step, because we need to obtain very clean plant for resist the condition of the climatic condition when you want to push in the land. Need to resist the high level of the sun radiation. So for that reason, this plant needs to have a very hard root. That's why it's the ounceing that produce that is used in the culture. Like I say, the most important is that we need to very clean plants. And thank you very much. I'm going to present my partner that is going to continue with the next step of the organogenesis of the sandia. Thank you very much.
Hi, continue with the exposition. This is the step number 4, that is the phase of acclimatization. Shoots are ready to acclimatize when roots have formed long, so less than 1 centimeter long. This process during 10 to 14 days. For acclimation, the vitro plants are removed from glaces and gently washed to remove root agar. Proceed to transplant multicellular trays with a sterile medium free of soil. For example, equal parts of mixed BX Pro and vermiculite. In this case, vermiculite is the sterile medium free of soil. These trays should be covered with transparent plastic and kept moist and light for 16 hours and 50 more. The plant should be covered for a week before gradually remove the plastic cover. In vitro morphogenesis of watermelon, cotyledon segments can be obtained from explants collected from the proximal region of tree. They all cotyledon in vitro germinated seedlings. on a culture media supplement with BAP, that is hormone and coconut water. In this case, now I explain one exercise that is a little experiment that is about the difference in the regenerative potential between different regions of watermelon cotyledon. The use of an appropriate explant warranty success in regeneration. Not all explants from the same plant or cotyledon have equal potential for regeneration. For example, the terminal part of cotyledon is better than other parts. For develop an efficient regeneration protocol, it is necessary to identify the best region plant extract where is necessary to identify the best region to extract the explant, because if you obtain the best part of the plant, it's more easy to cultivate this in a medium free of soil. The best source is the plant cotyledon extracted from seedlings germinated in vitro. However, not all regions of the cotyledon regeneration have equal potential. In this experiment, they have been explants from different regions of the cotyledon of watermelon to identify the best type of explant for ambitious bulb regeneration.
Okay, now we continue with the experiment.
Okay, the materials. We have agar, that is the medium where we're gonna do the experiment. Guys, I will put in your place some agar. Okay. The second material, and very important, is the watermelon and knife to cut the melon. So we proceed to cut the melon. Watermelon. It's very important to cut only the red zone of the watermelon in small pieces and put in the medium of agar. Now, the experiment consists in leave the medium with the watermelon pieces, and we need to take this and in some place that we can monitor the glasses and see how the pieces of watermelon are growing. Thank you very much, and this is our experiment. Any question for you?
So, after the time goes, we will get a plant in the
Yes.
experiment, and after that, it consists in the second step. That is, divide our cayus. In this, we will take another cup with agar, but this agar won't contain what is auxina. It will contain cytosina that promotes the sleep to grow up. So after that, we need to divide that cayus in another cup, and we will get more plants from one cayus. And now we present the plant that we divide, but the guys will divide this when that plant that we put now in the cube grows up to divide this in cayus. Now my partner will continue with the experiment.
Okay, yeah. There are one last step, and is the acclimatation. When we have the plant like this, we go to a greenhouse with the plant to get the acclimatation for the plant have the resistance to grow up in the soil.
Thank you.
Thank you. Now I'm going to give you the conclusion. As you can see, not all the region of watermelon cotyledon possess an equal regeneration potential. The students were culturing its plant from different regions of the watermelon to identify the best plant. We use as a source plant tissues culture concept in laboratory exercise 2nd edition. Thanks.
Today, plant tissue culture has been remarkably essential in agricultural biotechnology. It allows regeneration of a plant in an in vitro condition to occur. This process involves the differentiated cells to be differentiated into a callus, and redifferentiated back to an embryo that is capable to regenerate the whole plant. An example of tissue culture is micropropagation. It involves 6 plants growing in a media under sterile conditions. The plant rejuvenation can be done by 2 different ways, either through somatic embryogenesis or shoot culture. The final phase to complete a tissue culture is the transferring to soil and hardening of plantlet. The hardening process is indispensable in the in vitro plant to adapt to external environmental conditions. In this video, we are going to show step-by-step procedure of seed surface sterilization, germination and hardening process. Firstly, there is preparation of MS medium for seed germination, which are made up of macronutrient, micronutrient, Gambigs, B5 vitamin and FeNAEDTA. After mixing all ingredients, the half MSO liquid medium is then microwaved and autoclaved. After the solidified of culture agar medium, it is ready for inoculum. The standard procedure for sterilization technique includes: decant all the water inside the tube containing the seeds. Next, ethanol at 95% is added, followed by 20% bleach plus a drop of tween 20. Constant shaking for approximately 10 minutes is required to ensure complete sterilization. Seeds is then rinsed out with the autoclaved water and left dry. Then, seeds are inoculated into a conical flask containing the media. Afterwards, the flask containing the seeds are incubated in a well-lit area. After some period of incubation, seeds germination are to be expected. Further observation can be made on the morphology of the plant after few weeks later, ensuring it is ready for hardening. The hardening process is required to transfer in vitro plantlet to the ex vitro environment. With the successful of hardening, the establishment of plant which enable plantlets to grow up at fast rate. Next, the plantlets undergo acclimatization and adaptation period to mimic in vitro culture environment, such as covering plantlet in transparency of plastic bag to retain humidity and airflow, as well as placing shaded environment for low radiance and to avoid direct sunlight. After 4 weeks of observation period, the plantlet had survived an obvious growth development of height and number of leaves. It had grew approximately 1.7 centimeters and developed a newer leaves. There are a series of ethical issues that we need to take into considerations in terms of plant and tissues culture. One of the important points that we need to emphasize on is the inequality in the society where some individuals are not able to access to such technology. Other issues involves a competition between GMO and wild type and crossbreeding of variant. As for safety issues, there is a possibility that GMO may exhibit superior characteristics which may lead to allergic reaction. Thus, there may be a lack of genetic diversity in the population, and this will wipe out the whole population if there is a new disease outbreak. In social responsibility, agencies involved in monitoring GMO must have strict review upon all GMO in terms of potential environmental impacts and the risk of allergens. As successful of seed germination and plant hardening, it could provide business opportunity on the agricultural field of vegetable and fruit plantation, based on 6 characteristics. Mass production at shorter time spent is the main characteristics. Using this technique, higher ratio of in vitro culture flask can yield more seed germination than the equivalent area of soil. Products can also be harvested in shorter time as optimum and control conditions is set to get high yield of production. It can reduce land usage by using greenhouse. This saves space and land by designing multi-stories greenhouse compared to the conventional plantation in which is using monolayer of soil. With lesser land to be purchased and smaller area to maintain, cost is safe. Moreover, it is cheaper in cost. Purchase and usage of herbicides and pesticides avoid. The preparing of defined medium will be cheaper than purchasing fertile soil for every germination batch. Furthermore, the viability of plant grow will be higher and thus more product can be harvested for selling. In addition, it can produce variety of plant at a time by making partition in the greenhouse. As supplier, if there is more variety, buyer can just order and purchase straight from us without needed to go through other provider. This will create a win-win situation for both, as bulk purchase will be given discount and regular customer will have special offer too. Imported fruits can be planted locally too with this technique. Environmental conditions can be adjusted to mimic the condition of the origin country to suit the growth and production. Importation and transportation fees can be saved and thus cutting costs and increase earning. Health is another issue when choosing food to consume for consumer. The products will be organically produced and toxic-free. With this technique, we will able to provide not only cheaper food but also healthier diet to consumer by promoting organic food.